Hi everyone, in this video we will discuss few questions from TCA cycle. So the first question is discuss the reactions of TCA cycle and explain its amphibolic role. It's an essay question. So in case an essay question, just write the importance of this significance of this TCA cycle. So TCA cycle is the final common pathway for the oxidation of carbohydrate, lipid and protein. So all this metabolic compound produces acetyl-CoA as the end product, and this acetyl-CoA undergo complete oxidation in TCA cycle. That is the significance of TCA cycle. Then write about regarding where it takes place. So this reaction takes place within the mitochondria of the cell. Then draw the flow chart. Here you should mention about the substrate, product, enzyme and coenzyme molecule. Important enzymes in case of TCA cycles are: one is isocitrate dehydrogenase, which produces alpha ketoglutarate from isocitrate, and this reaction releases one molecule of NADH. And the second important enzyme is alpha ketoglutarate dehydrogenase. This reaction also produces one molecule of NADH. And the third important enzyme is succinate thiokinase, which produces succinate from succinyl-CoA, and is an example for substrate level phosphorylation where one molecule of GTP is produced from GDP. Another important enzyme is succinate dehydrogenase enzyme, and this reaction produces one molecule of FADH2. And another important enzyme in TCA cycle, that is the last enzyme, that is malate dehydrogenase enzyme, and this reaction regenerate oxaloacetate, and this reaction also produces one molecule of NADH. And just then you should write about the energetics of TCA cycle. So one cycle of this TCA pathway produces 3 molecules of NADH, 1 molecule of FADH2 and 1 GTP molecule. So one cycle of this TCA pathway, TCA cycle produces 10 ATP. And another important questions related to TCA cycle: what are the inhibitors of TCA cycle? So just try to learn at least 2 examples. What is the inhibitory molecule and which enzyme is inhibited, and what is the type of inhibition? So fluoroacetate inhibit aconitase enzyme of the TCA cycle. It is a type of suicidal inhibition. And the second inhibitor is arsenate, which inhibit alpha ketoglutarate dehydrogenase. It is a type of non-competitive inhibition. And malonate molecule inhibiting succinate dehydrogenase enzyme. It is an example for competitive type of enzyme inhibition. So these are the inhibitors of TCA cycle. And it's a 1 mark question. Name 2 carbon dioxide releasing steps in TCA cycle. So there are 2 reactions in TCA cycle which produces carbon dioxide. One is the reaction mediated by isocitrate dehydrogenase, and second one is reaction mediated by alpha ketoglutarate dehydrogenase. And mention about the reaction. And another important thing related to TCA cycle, one is its amphibolic role, and second one is its anaplerotic role. So what is amphibolic role? You can expect short answer type questions from this topic: amphibolic role as well as anaplerotic role. So what is the amphibolic function of TCA cycle? Amphibolic function means both anabolic function and catabolic function. So what is the catabolic function of this TCA cycle? So TCA cycle is the final common pathway for the catabolism of carbohydrate, protein and lipid. So that is the catabolic function of this TCA cycle. Then what is the anabolic function of TCA cycle? Anabolic means synthetic pathway. So this TCA cycle intermediates are used for the biosynthesis of other compound. For example, oxaloacetate is used for the synthesis of glucose, that is during gluconeogenesis, and citrate is used for the biosynthesis of fatty acid, and alpha ketoglutarate, which is used for purine biosynthesis, and succinyl-CoA, it is used for heme biosynthesis. So these are the anabolic functions of TCA cycle. So TCA cycle has both anabolic function and catabolic function, thereby the TCA cycle is said to have amphibolic role. The second thing is, what is anaplerotic reaction? That is also important. What are the anaplerotic reactions of TCA cycle? So as I mentioned earlier, TCA cycle intermediates are used for biosynthetic pathway. So these biosynthetic pathway, once they are used for biosynthetic pathway, that can deplete the amount or concentration of this TCA cycle in For that purpose, this intermediate has to be regenerated. So there are some reactions in our body where this TCA cycle intermediates are continuously regenerated. That will refill or that can replenish this TCA cycle intermediates. For example, oxaloacetate can be produced from pyruvate by the enzyme pyruvate carboxylase, and malate can be produced from pyruvate by malic enzyme. And oxaloacetate is an intermediate in TCA cycle which can be produced from aspartate. It's an amino acid with the help of aspartate transaminase enzyme. It's a transamination reaction. And alpha ketoglutarate can be produced from glutamate by another transamination reaction. So these are the example for anaplerotic reactions. So that's all regarding TCA cycle. Thank you.